In a previous lesson, we learned about what's happening to this mouse in the bottom part of the diagram here. We're taking it from its home chamber, putting it into a training context, the red chamber, and then shocking it on its foot. So it's going to have an experience, it's going to learn something. It will associate the red chamber with the shock. And we put it back in its home chamber to rest. And then when we just put it back into that training chamber without shocking it, it will show fear. So that's the testing context. It's going to show fear because it learned to associate the red chamber with the foot shock. And then these pictures up here represented what's happening in the brain of the animal. And we could describe it as an encoding stage. The brain is responding to the learning event. A storage phase where neurons are growing stronger synapses with each other. We called them engram neurons. And then the retrieval of the fear memory was that those engram neurons get reactivated by the testing context, the red chamber. In this lesson, then, we'll see how it is that optogenetics provides strong evidence for this story, for the memory formation process. First, let's remind ourselves that the way neurons operate is that they activate each other at the locations of these synapses. Neurons do not respond to light. But the technique of optogenetics seeks to change that. What optogenetics does is it engineers neurons to respond to light. So here we have our cartoon neurons here, and notice there's this piece of DNA. Now DNA is the hereditary molecule for life on Earth, and we would say a section of DNA that codes for a protein is a gene. So this gene was taken from another species that makes this protein that responds to light. It's a protein channel. So by inserting the DNA from another species into the neurons of the mouse, you've created a mouse with brain cells that will be able to respond to light. So here we have a mouse neuron that was genetically modified to express a protein that they don't normally express. And we can see what these special protein channels are. Well, they're like other protein channels that allow charged particles called ions into the cell, and that has the effect of activating the neuron. But they only open up when exposed to blue light. So when blue light is shining on these protein channels, they open up and allow charged particles called ions into the neuron, and that's what triggers it to generate its output signal. So these are light-activated channels, and they get opened up when blue light is shining on them. Notice, it doesn't matter what kind of electrical activity is out here on the dendrites. In fact, there may be no activity here, but if you shine blue light on this neuron, it will generate an electrical signal. So now we have a neuron that will respond to light, in this case, blue light. So back to our diagram here, we've inserted DNA into these mouse neurons, so they're genetically engineered neurons. And now, when the animal goes ahead and we put it into the training context and it gets a shock, remember some of those neurons are going to be activated by the learning event. Well, it's these neurons that get activated that will begin to express that special protein. So it's only the activated neurons that install these light-activated channels. So the training activated neurons make this special channel, so we could call these the opsin tagged neurons. Opsin is the name of that protein channel. We're tagging those neurons that were activated by the learning event, and now these neurons are installing a new protein channel that can be activated by light. So when we shine blue light on these particular neurons, it will turn them on, no matter what kind of input activity they're getting. So in this way, scientists can now activate these neurons at will. They have control of them. In this picture here, we see a rat with an optical fiber inserted into its brain at a certain location. And so light from that fiber will illuminate a localized population of neurons. Down here, we see a sort of a cartoon neuron. We see the channels that have been inserted into the membrane and blue light hitting the neuron. And so scientists can activate this neuron just by shining blue light on it. So with optogenetics, neurons can be electrically excited by light. The light-sensitive channels activate the neuron only when the blue light is on. Engineering the neurons with different channels allows scientists to silence the neuron with yellow light. So inserting a different gene for a different protein channel that responds to yellow light, that will allow scientists to inhibit a neuron when they shine the yellow light on them. We could say silence the neuron. So with a certain kind of channel, blue light will activate the neuron. With a different kind of channel, yellow light will inhibit the neuron, will silence the neuron. So now we have a population of neurons, depending on what channels were inserted into those neurons, but they can be controlled by blue or yellow light. Again, the blue light will activate the neurons if you've engineered them with that particular channel, but if you've engineered them with that other kind of channel, yellow light will silence these neurons. But notice, it's only the neurons that were activated by the learning event that now express these light-activated channels. So this allows scientists to have complete control over the very neurons involved in a learning event and the subsequent formation of a memory trace. All right, with this significant background, we're ready to see the payoff of this optogenetics technique and how it can be used to directly gain control of the engram neurons to demonstrate that, in fact, memories are formed by the formation of a set of engram neurons and memories are retrieved by the reactivation of those engram neurons. Here we go. So up top here, we have an animal in the training context. It's learning to associate the red chamber with the foot shock. So a set of neurons was activated by that learning, and now using optogenetics, they were genetically engineered to now express a protein channel that is activated by yellow light. But notice, we're going to take the animal, we're going to put it into, we'll give it some rest, but then we'll put it back into the red training context. A normal mouse would show fear because it learned to associate the red chamber with the foot shock. But look, this animal is not freezing. Why is that? Because at the very moment they put it back into the red chamber, scientists turned on the yellow light, shined yellow light on the engram neurons, and that's going to silence them. So when scientists silence these engram neurons, the animal cannot retrieve the fear memory. What this shows is that activation of engram neurons is necessary for memory expression, the retrieval of the memory. If you block the activity of those engram neurons, the animal cannot retrieve the fear to that training context. Consider another nice example of using optogenetics to study memory. Here we have the animal in its red training context getting the foot shock. So we're going to have a population of neurons activated, and again, they were engineered, so now they're going to express these light-activated channels, but this time they're activated by blue light. Now notice we're going to take this animal, put it back into its home chamber, a neutral context. It never learned to fear anything in its home chamber. And notice it is fearful, it's freezing, it's showing fear. Why is that? Because scientists were now shining blue light on those engram neurons and reactivating these engram neurons in a new environment, in a new context. So the fear memory is being expressed in a new context because scientists just reactivated those engram neurons with the blue light. This illustrates that activation of engram neurons is sufficient for memory expression, in this case retrieval. So controlling neurons with optogenetics allows scientists to directly study the process of memory formation. You can have an animal that has formed a memory, but then you can block the expression of that memory by silencing the engram neurons. You can have an animal that formed a fear memory and then make it show that fear in a totally new context by reactivating the engram neurons. You can literally control the very neurons involved in the formation of a memory trace and whose activity represents the retrieval of a memory. So optogenetics research provided strong evidence for this process of memory formation that we illustrated in the previous lesson. The encoding stage, the storage stage, the reactivation stage. It is optogenetics that gave us direct evidence of the story shown in this illustration.